This video is part of the lecture Plant Breeding in a Modern World in the scope of the course Applied Genetics at the University of Ostrava. Previously, we showed how plants can be genetically engineered by inserting foreign DNA in their genome. The inserted DNA was either from the same species, in which the process is called cisgenesis, or from another species, for transgenesis. Another way to genetically engineer a plant or any organism was developed at the beginning of the 21st century: gene editing, which is also called subgenesis. Contrary to cis and transgenesis, subgenesis does not introduce any foreign DNA in the genome of the modified plant. Rather, it induces the organism to make its own guided modification of its DNA. There are different technologies used for gene editing, but the three most exploited ones are TALEN, zinc finger nuclease, and the CRISPR-Cas9 complex. Although they all have their own specificities, each technology is characterized by two main components. The first one recognizes and binds to a specific DNA sequence in the genome that we wish to modify. The second one is an endonuclease which cleaves the DNA at a specific recognition place. Alternatively, after the successful detection and cleavage of the DNA sequence of interest, the cell repair mechanism can be guided by added DNA fragments. The fragments pair up with the damaged genomic DNA because they have complementary end sequences. Then the cell's repairing mechanism mends the broken strand using the added DNA fragments as a template. This creates a new, engineered sequence in the plant genome. Genome editing represents a few advantages to classic genetic engineering techniques. From a technical and biological point of view, the risk of undesired genetic modification because of the unwanted insertion in a coding or regulating position in the genome is minimal. In agrobacterium-mediated genetic engineering, such occurrences are highly likely because the restriction sites are only 5 to 6 base pairs long, so the same sequence motif can be found throughout the genome. Gene editing technologies are based on the recognition of a DNA sequence of at least 15 and more often 20 base pair long. The probability of having a unique DNA sequence of that length more than once in the entire genome is very low, allowing for the precise editing of a desired gene or other types of DNA sequences. Moreover, contrary to genetic engineering with mechanical insertions, where only a tiny fraction of the treated cells will be successfully and permanently transformed, the success rate of genome editing are remarkably higher, with some studies reporting of up to 14% of successful hereditary targeted edits of the genome. Finally, because no foreign DNA is inserted in the plant genome, crops with edited genome are not subject to regulations that apply to classical GMO crops obtained by cis or transgenesis. Edited crops thus require less safety testing in order to be deemed safe for consumption, which significantly decreases the delays between their creation and commercialization. Tomatoes are one of the fruits-producing crops that have been the most frequently modified with genome editing so far. For instance, an enzyme produced by the gene PSY1 is responsible for producing the red color of tomatoes by converting orange carotenoids in red pigments. This gene was disabled using CRISPR genome editing, resulting in the accumulation of carotenoid pigments in the tomato fruit and orange-colored tomatoes, which are in high demand in the Asian market. Other allele editing has produced pink-colored tomatoes, which are popular in Europe, slow-ripening tomatoes that can stay on supermarket shelves for up to 40 days, and drought-resistant tomato plants. Even more interestingly, genome editing was used for a de novo domestication of the tomato. By simply disabling 6 genes in wild tomatoes, a new variety that has determinate growth, more flowers per fruit, larger fruits, and higher lycopene content of the fruit was created within a year. This is a significantly faster domestication time than the thousands of years required for the original domestication of the tomatoes done with mass selection.